This lecture is the first part in a series of lectures on SARS-CoV-2 and COVID-19 for Micro 320, Emerging Infectious Diseases. So the first question, what is COVID-19? By now, we're all pretty familiar with this. The 2019 novel coronavirus infection is a respiratory infection caused by the virus SARS-CoV-2. The word corona, from the Greek word for crown, I'm Greek, so I got to point that out, is illustrated here. You can see the virus and its various spikes giving it that corona or crown-like appearance. It was first identified December 2019 in Wuhan, China, but has obviously spread globally since then. There's a fair amount of question as to where exactly the virus originated, whether it was a cave in Yunnan province or the Wuhan Institute of Virology, for example. And so these are ongoing debates and discussions, and I'll address those in a future lecture. The coronavirus taxonomy illustrates that there are quite a few members of the genus beta coronavirus that are pretty closely related to SARS-CoV-2. The alpha coronaviruses that you see here consist of a variety of cold-causing or human cold-causing viruses such as 229E and NL63. These viruses use an aminopeptidase N receptor on the host cell surface, whereas the beta coronaviruses look for the ACE2 receptor. And the ACE2 is part of the renin-angiotensin system that's responsible for managing blood pressure, for example. So you see a couple cold-causing coronaviruses such as HKU1 and OC43, as well as some of the more familiar coronaviruses, SARS-CoV-1, which caused SARS in the early 2000s, and then MERS-CoV, which is a very deadly version of coronavirus that presumably jumped from camels to humans and periodically does so. And then, of course, SARS-CoV-2, the etiologic agent of COVID-19. These common cold-causing viruses, OC43, NL63, HKU1, 229E, have been documented to cause colds that have led to deaths, but there's no reliable statistics on the actual numbers. But these viruses circulate every year. And it's presumed that eventually SARS-CoV-2 will join them as just an endemic virus that largely passes through the population in a seasonal way. So just a bit about the ultrastructure of the virus. Here's a picture of the virus showing envelope proteins, the spike protein, which I alluded to before, giving the corona-like appearance. And these spike proteins, I should point out, are also the target for vaccines. So the messenger RNA vaccines, for example, encode a piece of messenger RNA, contain a piece of messenger RNA, which encode the instructions for producing these spike proteins, which in turn would elicit an antibody response. These membrane proteins are present, as well as this hemagglutinin esterase protein, and then, of course, nucleoproteins that intimately associate with the viral RNA to protect it from degradation and keep it in a certain conformation. And then, of course, the genomic RNA that's buried within there. So I've just got some text here to explain what some of these things do. So S, for example, forms the prominent spikes. It binds to the ACE2 receptor. ACE2 is found on human respiratory tract and gut tract epithelium, which again illustrates how this virus can actually infect all throughout the human respiratory tract and can even presumably infect the gut tract. And it's, again, one of the reasons why we find this organism or are able to find this organism in fecal contents. We can use the feces of individuals to track virus prevalence and even track virus transmission in the community. The gene for the spike protein in SARS-CoV-2 has an insertion of 12 bases, and this gives it what's called a furin cleavage site. And this furin, which I'll talk about, is an additional processing step that might give the virus much more efficient ability to infect human cells. And so again, I'll talk a bit more about this when I get into virus origins. And as I've already alluded to, S is a prominent vaccine target. E and M are needed for envelope assembly and budding. They're recruited to the ergic, which is the endoplasmic reticulum, and Golgi intermediate compartments. And this is all about trafficking the virus, which I'll talk about. E also forms ion channels, and that's probably what helps in these budding processes, these intracellular budding processes. The HE, the hemagglutinin allows for sialic acid receptor binding. And so presumably then, as I'll show you in a subsequent slide, the virus initially can make contact with abundant sialic acid residues on the host cell and then switch from sialic acid binding to protein binding. And it presumably can also, as an esterase, can cleave these as well so that it doesn't get stuck there on the membrane surface. The N, or nucleocapsid protein, as I said, is essential for packaging and protection of the virus RNA. But another interesting feature is the potential for it to facilitate immune evasion. In this particular case, interferon suppression. Interferon is an alarm protein that's produced by infected cells, basically to alert neighboring cells that have not yet been infected that basically I'm infected and something's coming, and so the interferon response will then allow those cells to essentially batten down the hatches before the viruses get to it. So this is just a little look at amino acid sequences and using these amino acid sequences, specifically of the receptor binding domains of different viruses, to understand coronavirus relatedness. And so again, you can see where bits of the receptor binding domain, and again, the receptor binding domain allows the virus to bind to ACE2. These particular viruses to bind to ACE2. And so where you see yellow, these are highly conserved amino acids in this particular amino acid sequence. And then these cyan indicators show variable regions. And so you can see there's quite a lot of variable regions between these different viruses: SARS-CoV-2, SARS-CoV-1, and MERS. Down here is just another version of this experiment so we can get a sense of evolutionary relatedness. So here is just a protein gel showing you the receptor binding domain proteins run on a gel so you can see the banding. And here's a Western blot in which the isolated proteins have been run on a gel and then stained with specific antibodies, in this case SARS-CoV-1 receptor binding domain specific antibodies. And you can see there is cross-reaction between the SARS-CoV-1 receptor binding domain antibodies and itself, not surprising, and the antibodies with SARS-CoV-2 receptor binding domain. Conversely, only the MERS-CoV receptor binding domain specific antibodies only bind to themselves and not the other two SARS viruses. And so that, again, allows us to estimate viral relationship. This next slide takes a close look at the functional domains in the spike proteins of SARS-CoV-2 and SARS-CoV-1, and then there's amino acid sequences highlighting some really important sites within the spike protein. So S1 that you see here in this spike protein is contains the ACE2 receptor binding domain, as you see here. S2 consists of primarily of the fusion peptide, which facilitates membrane fusion, allowing the virus to actually enter a susceptible host cell. But in between these domains, S1 and S2, are these cleavage sites. For example, for SARS-CoV-2, the furin and TMPR SS2, this transmembrane serine protease, and trypsin proteases that can cleave at various sites. And again, the purpose of that cleavage is to cut here and expose the fusion peptide so the virus can actually get into the susceptible host cell. SARS-CoV-1 has cleavage sites as well, the trypsin and the transmembrane serine protease, but lacks the furin protease receptor site. And you can see that here, the PRRAR, this region here, is presumably the furin cleavage site. And again, SARS-CoV-1, its close relative lacks that particular site, but does have this key arginine here. in order to facilitate cleavage. So both viruses can obviously get into cells still, even though SARS-CoV-2 probably does so more efficiently by having this extra piece of amino acid sequence. This is a map of the subgenomic RNAs and gives you a sense of the replication process in 3 stages. So the cell is infected, and initially the virus will encode the ORF1A polyprotein, which its primary goal is to hijack the cell. And by hijack the cell, we're talking, securing ribosomes so that the ribosomes of the host are exclusively dedicated to making virus proteins. But it's also about disrupting innate immunity such that the cell and the human cannot protect itself. And so and again, part of the innate immune response is, for example, knocking out the interferon response. And so the ORF1A polyprotein is made, and then there's a programmed ribosomal frame shift signal so that 25% of the ribosomes will slip past this ORF1A and begin making ORF1B, which contains the RNA-dependent RNA polymerase. The RNA polymerase then will start transcribing RNA to make subgenomic RNAs as well as full-length genomic RNAs. And this information can actually feed back and accelerate the hijacking process. But these subgenomic and genomic RNAs can be used to ultimately to make fully formed viruses. The subgenomic RNAs make structural proteins that the virus needs to build itself, and then hunks of the genomic RNA must be packaged, and then ultimately that facilitates assembly and release. So in this slide, we get a look at the full-length organization of the genome, ORF1A, ORF1B, and then these various proteins here. After primary translation, we have the main polyprotein, polyprotein 1A and polyprotein 1AB, broken into the non-structural proteins, so you can kind of see where they fall. NSP1, NSP2, NSP3, and so forth. Then the back region here of this roughly 30,000 base pair genomic RNA fragment encodes these subgenomic RNAs that you see here. Some of these are non-structural proteins, and some of them are structural proteins, such as E and M and N and so forth, which I've talked about. One of the important aspects of this, though, is this method of translation where large polyproteins are made, is the need to cut these. And so some of these non-structural proteins, such as you see here, NSP3 and then NSP5, make proteases. So the NSP3 protease, PL protease, cleaves here and clips off NSP1 and NSP2, as well as the bits of itself, so that it can function more efficiently. Whereas the 3CL protease that you see here, that's encoded in this essentially NSP5 region, will cut in these particular locations to liberate these non-structural proteins and allow them to do their particular job, particularly, for example, the RNA-dependent RNA polymerase, the helicase, and so forth, which I'll talk about. The subgenomic RNAs that you see here, as I said, here's this bit of subgenomic RNA encodes the spike, and then here's the envelope protein and membrane protein and the nucleocapsid protein that I've mentioned before. The additional non-structural proteins include ORF3A and ORF6. The ORF stands for open reading frame, so these are just basically long reads that produce a functional protein. ORF6, ORF7A and 7B, and then ORF8, and lastly ORF9B. I should also point out this little note here, the leaky scanning that you see here. It's basically there's a weak initiation codon in the messenger RNA that's sometimes skipped by the ribosome so that you can get either the nucleocapsid protein or the ORF9B subgenomic fragment. So here's a rundown of some of the functions of non-structural proteins. A lot of this is not a lot of it. Some of it's preliminary and hypothetical at this point. But so, for example, NSP1 likely inhibits host translation by interacting with the 40S ribosomal subunit. So the 40S ribosomal subunit is the small subunit in eukaryotic or human host cells. And as a result of that interaction, it basically facilitates sort of a preferential translation of virus proteins. The NSP2 most likely arrests normal cell functions. And again, the idea is to do essentially what NSP1 is doing to force the cell to preferentially read virus information, virus RNA particularly. NSP3 is the protease that I mentioned previously, the individual PL protease. NSP4 assists in the formation of double membrane cytoplasmic vesicles, which again becomes really important in the intracellular trafficking of the virus and ultimately in its assembly. NSP5 is the CL protease that I showed previously. NSP6 probably works with NSP4, also facilitating the formation of double membrane cytoplasmic vesicles. NSP7 and 8, which I don't have on here, but are most likely cofactors in the RNA-dependent RNA polymerase, allowing it to work efficiently in replicating the virus RNA genome. And then NSP9 is a single-stranded binding protein. So again, to bind to the single-stranded RNA, preventing it in a sense from forming stem loops and so forth, so that it can be efficiently replicated. NSP12 is the actual virus RNA polymerase. So that's what actually copies the virus RNA, and it's also, no surprise, a great antiviral target. Presumably, what remdesivir, for example, would attempt to block. And NSP13 is a virus helicase, which helps unwind the RNA for replication. So any regions of double-strandedness, for example, or anything like that, so the virus RNA can be unwound and read efficiently. NSP14 is a proofreading, has a proofreading function, so it's 3', 5' exoribonuclease activity, and therefore can actually, as the polymerase is laying down bases, this NSP14 can facilitate going backwards and basically cutting out any errors, because the virus is a very, very big genome. And if it sustains too many errors, too many mutations, it's quite possible that particular strain of virus would go extinct. And so therefore having a proofreading function is essential. But still, the proofreading's not so great, right? Because we still do get a sufficient number of variants that have allowed the virus to continue making comebacks, such as what we're seeing now with the Delta variant. NSP15 chops up viral RNA to essentially cover its tracks. And this is an important feature because the virus, if the virus's RNA builds up, that can actually trigger an immune response, essentially, an innate immune response. And our cells have a way of detecting double-stranded and viral RNA in the cell and activating antiviral defenses. So again, covering its tracks is an important adaptation. And of course, NSP16 would cap, adding a messenger RNA cap to its genomic RNA, basically allowing it to look like mature messenger RNA in the mature host messenger RNA. And that cap is essential for allowing it to be translated into proteins. One interesting feature of virus replication is this concept of discontinuous transcription. So again, you can see the full-length virus messenger RNA that pictured here. It's got a 5' cap and a 3' poly A tail. And again, that allows it to behave like a mature eukaryotic or animal cell messenger RNA. But one of the things that as I've mentioned, is there's this concept of discontinuous transcription, where you have the first process is the production. Here's our genomic RNA pictured here, and within the genomic RNA is a leader. These leader transcription regulating sequences, as well as body transcription regulating sequences, pictured with these little red rectangles. And so again, the polymerase would touch down here, and essentially would touch down here and essentially read this into minus RNA transcription. So we'd go from a plus RNA genome, which is then transcribed into a minus strand, essentially. And so here's our minus strand, full length. You can make full length minus strands, but a lot of these strands, because the virus doesn't want to just make genomic RNA, as pictured here, you make some subgenomic RNAs and some genomic RNAs. The subgenomic RNAs are used to make structural proteins, and the genomic RNAs are packaged in order to make complete virus particles. So some full-length genomes are made, as you see down here. The polymerase doesn't jump, and you get a full genomic strand. Where in these cases, the first TRS, the first body TRS is encountered by the polymerase, and that causes it to then make a small bit of the RNA and then jump with its nascent RNA to the leader TRS and then complete transcription. And so you end up with this little piece here, for example. And then here's another example where the polymerase jumps at the second TRS. So then you make ORF8, for example. So then the polymerase touches down and reads through to the second TRS and then jumps to the leader TRS. And so again, you end up with this, and so on and so forth. So again, the virus, based on this discontinuous transcription approach, is making both subgenomic and full-length genomic RNAs. And again, this obviously adds major deletions in these subgenomic RNAs, such that you don't have to waste energy making all this meat when you don't need it. All you need is these subgenomics, again, so the virus can make the bits and pieces that it needs for structure and so forth. This slide is just an overview of some of the accessory proteins that SARS-CoV-2 genome encodes. So at the top of the list here is ORF3A, which inserts into the endoplasmic reticulum membrane and forms a pore, which presumably aids in budding throughout those intracellular compartments. But ORF3A is also known to induce NF-kappa B, for example, the transcription factor that leads to an excess production of pro-inflammatory interleukin 1B, as well as activating tumor necrosis factor alpha. These things contribute to the so-called cytokine storm, which results in a lot of the damage that is typical of bad outcomes with COVID-19. You can also see this other mechanism here in which the virus infects cells of the upper respiratory tract and triggers the P2RX7 receptor here to activate the NLRP3 inflammasome. And so again, these virus-infected cells will produce cytokines, but they also produce an abundance of ATP, and this receptor then recognizes that stress response and activates the inflammasome, which also results in interleukin 1B, which in turn, and interleukin 18. And so as a result of that, there's an overstimulation of immune system cells and as well as epithelial cells to produce additional cytokines and come to the fight. And by doing that, these cells elicit collateral damage. And so again, you can see the cytokine storm mediators, including tumor necrosis factor alpha, and in turn leading to ARDS, acute respiratory distress syndrome, systemic inflammatory response, because a lot of these is directed towards damaging the endothelium of blood vessels. There's shock and multi-organ dysfunction as a result of coagulopathy, so a lot of clotting that can happen that can block off capillaries, for example, and choke off oxygen to various organs. ORF3b, N, ORF6, ORF9b all contribute to blocking interferon, part of the innate immune response. So again, interferon is that alarm chemical that infected cells will produce to alert neighboring cells that, I'm infected. Again, batten down the hatches and prepare for this, for what's coming. And usually what that means is that the cells are then primed to produce nucleases, RNA nucleases, for example, that would then degrade the incoming virus RNA. ORF7a might prevent virions from being bound to the membrane surface by tethering, essentially. And so I'll show you a mechanism for that. So again, the viruses have to leave the cell, but if they're stuck to the cell as they're exiting, it's essentially like a boat being moored to a dock and then having to unmoor yourself in order for the boat to be able to drive away. And so here's a overview of the entry and spread. The virus that you see here, SARS-CoV-2, has its little spike proteins, and these spike proteins, in addition to the hemagglutinins that I'd shown you previously, are working together to bind to the epithelial cell surface. And so the primary thing that happens at first is weak interaction with sialic acids, which again are abundant on host cells. And as these viruses concentrate on the surface, that starts to allow more and more of these surface spike and protein receptor interactions. And as these tighter and more efficient binding events take place, surface proteases that the host cell produces can then process the spike proteins and facilitate this membrane fusion event that you see here. And ultimately, the virus can then replicate and ultimately butt its way out of the cell, as pictured here. So there's a couple different ways for spread to happen. You have this normal virus approach, I don't hate to say normal, but typical canonical spread, which is budding from the surface into the outside world, and then this fully formed virus particle can then bind to a susceptible host cell. But there's another approach that SARS-CoV-2 and other viruses use, and that's syncytia formation. And syncytia formation occurs when virus spike proteins, which are being assembled and displayed on the surface of the infected host cell. This is actually an important process because displaying antigens like this tells the immune response, I'm infected, basically kill me. But in this particular case, it leads to syncytia formation. So these syncytia, this is a neighboring cell, and the neighboring cell's sialic acids can interact with the spike proteins, the hemagglutinin and spike proteins on the that are displayed on the surface of the infected cell, and that causes membrane fusion to occur. So you see these places here where the cytoplasms are now continuous. That's basically just providing a conduit so that virus RNA can actually pass into this neighboring cell and stimulate replication. So the virus doesn't need to make a fully formed particle that goes out into the world. The virus can simply spread its RNA from cell to cell through the formation of these syncytia. So this slide is just showing you a side-by-side look at SARS-CoV-1 and SARS-CoV-2 processing. So over here on the left, and again, you can look at the legend, the SARS-CoV-1 virus is using both the transmembrane serine protease as well as a trypsin protease in order to process its spike proteins to facilitate membrane fusion and internalization, ultimately trafficking through the endoplasmic reticulum and Golgi to then be released into the world. So they can also use cathepsin, which would occur in these vesicles, these intracellular vesicles. SARS-CoV-2 is a little different because not only does it use trypsin and the transmembrane serine protease, but it also can use the furin protease. So again, SARS-CoV-1 lacks that efficient furin protease cleavage site in its spike protein, as I showed you previously. So having these different methods of spike protein maturation presumably gives SARS-CoV-2 far more efficient spread, internalization and spread. And so again, the virus enters through fusion, releases its RNA. The RNA is replicated and translated, and at various stages, another important thing to see here is that it's encountering furin proteases intracellularly through the endoplasmic reticulum and Golgi, which then allow it to process intracellularly, ultimately to then bud out into the world. So again, having this extra cleavage mechanism presumably makes the virus far more efficient. So we know that furin cleavage of SARS-CoV-2 promotes efficient spread. It's not essential for it, meaning that if you remove that furin protease from a cell, the virus will still operate, but it's not as efficient. And so therefore it's a very important evolutionary strategy. How did SARS-CoV-2 evolve this furin protease capability, those 12 base pairs that I had alluded to previously? So this slide is just an overview of the virus replication mechanisms and brings it all together. Here in step 1, you see SARS-CoV-2 recognizing the ACE2 receptor in the presence of this transmembrane serine protease will cleave that receptor to facilitate tight interaction and exposure of the fusion peptide. Now remember that furin protease, also on the surface, also facilitates this cleavage, and also trypsin. So other proteases can do this as well. But again, having that additional capacity, the furin cleavage site, for example, enhances the efficiency of this process. So by exposing the fusion peptide, you have this membrane fusion event, which allows the virus to enter. The virus then releases its plus-stranded messenger RNA into the cytoplasm, where it can be translated, and ultimately the polyproteins that are produced have to be cleaved by those proteases that I discussed. And so here are the individual proteins that result from this. The full-length plus-stranded mRNA can also be transcribed into minus strands, which in turn can be transcribed into additional genomes, full-length genomes, and the subgenomic RNAs, which again can be translated into structural proteins and those accessory proteins. Here are some of the structural proteins embedded in the endoplasmic reticulum membrane. So again, membrane protein, envelope protein, spike protein, and so forth, ready to be trafficked through the ERGIC, the endoplasmic reticulum Golgi intermediate compartment, they are trafficked to the Golgi, and then ultimately assembled and budded off of the Golgi, and then they mature in these budded vesicles, and then they form these particles which can then be exocytosed into the outside world. Once these viruses are exocytosed, they can then be encountered by antigen-presenting cells, cells of the immune response, such as macrophages. And so here's that process happening where this particular APC has a receptor that can recognize the virus, and then that stimulates the APC then to release cytokines. And in some cases, especially individuals with severe comorbidities, you have an enhanced pro-inflammatory response, which results in, again, the bad outcomes of COVID-19, including ARDS. The other thing that's not shown here is that the virus can also elicit the formation of syncytia, which I've talked about, these sort of membrane fusion events between adjacent host cells, allowing full-length genomes to simply pass through without forming a fully formed particle. And here we see the last step in the virus replication cycle, actually leaving the host cell. And so in this particular instance, the virus is assembled at the cytoplasmic membrane surface. It's embedded its spikes, and it's starting to form a bud in this particular case. And eventually will form the fully formed particle, which has exited the cell. But the tether endimers that you see here in purple are induced by interferon to trap these viruses. So again, interferon is part of the innate immune response, and clearly this is an evolutionary adaptation to preventing virus spread within the body. And as you can see, a variety of viruses like Ebola and SARS and HIV have come up with strategies for circumventing this tether endimer formation. Again, this is like, attaching a boat to a dock through a mooring line, having to release that mooring line, as I talked about before. And so these proteins will actually block the expression of tetherin so that the virus can leave. If this doesn't happen, if there's a defect, for example, in ORF7A, this tetherin interaction will take place, fix the virus to the surface, and eventually the virus will be endocytosed and degraded. So this concludes the basic virology lecture for SARS-CoV-2 and COVID-19. The subsequent lectures will cover epidemiology of the virus, treatments, vaccines, and so forth.